Hello everybody, my name is Catherine. Today I will present some of the preliminary results of a big project of DNR barcode that we are doing here at San Marcos University. I will present the preliminary DNR barcoding analysis of the Entomofauna of Yarinacocha Lake. The Yarinacocha Lake is part of the Ucayali River Basin in the Amazon region of Peru, which is characterized by a warm climate and high levels of precipitation. It belongs to the tropical rainforest limestone, and it's approximately 20 kilometers long. The principal objective of the project was to generate DNA barcodes of Entomofauna of the Yarinacocha Lake. Here we have a little map of the sample points in the lake. For the sample processing, we used two sample methods, qualitative and quantitative. Both were applied in the lake and in surrounding ecosystems. We used a 500 micrometers mesh opening DNet and household strainers. And then the organisms collected were fixed and stored in 96 degrees ethanol. For the molecular analysis, all DNA extractions were performed using the Promega extraction kit, and the amplification of the cogen was carried out with Zetaplank primers, and both strands were sequenced by Macrogene Korea. Sequences obtained were checked against the GenBank and Ball system databases. For the results, we have that in all cases, all sequences match the a priori identified orders. So we had one Ephemeroptera, one Adonata, 3 Hemiptera, 2 Coleoptera, and 1 Diptera. 2 of them resulted with high similarity at the species level. Those were Perithemys parzifalis and Bonoa omnigenus. For the other morph species, the genus Bolostoma was identified, and we also corroborated some families. Those were Petidae, Dictiscid, Noteridae, and Chironomid. A sequence of the order Hemiptera was found to be basal to Omphacoridae and Miridae in the both Taxonidae tree, differing from the priori morphological identification. Here we have some pictures of the specimens collected. On top, on the left side, we have Perithemys parzifalis, which is in nymph phase. On the right side, we have Bonoa omnigenus, which is in adult phase. Down there, we have Belostoma on the left side, which is in nymph phase, and Bethidae, which is in the same phase. Then down there, we have Noteridae in the left side, which is in adult phase. And on the right side, we have Chironomid, which is in larval phase. Down there, we have Dictiscid on the left side and Hemiptera on the right side. This one was the specimen that was primarily identified as Coryxidae, but when checked against the databases, didn't match the same family. To conclude, we have that our results would constitute the first co-sequences for 6 of the 8 tags analyzed in the present study that would enrich the reference library for environmental DNA studies at the Yarinacocha Lake. We also want to thank to the Faculty of Biology of the University of San Marcos and the Museum of the Same, On the Seat, and the World Bank for all the help and funding. Thank you so much for listening.